Hello and welcome to this video on the compass of blots used in biochemistry, molecular biology, forensics and more. When you see this sort of image, your first thought is probably some variation on "I know what that is" or "what is that?" In the latter case, this video will tell you what it might be. In the former, the answer might vary when asked what it is. The variation is a result of there being technically 4 but commonly 3 different kinds of blots used, and here you're seeing one of them. The general underlying and common theme to all blotting techniques is as follows. You have a semi-permeable structure, and this is made of something that is like a filter membrane, but not a very good filter. Just about everything can pass through it, but they do so at different speeds. This leads to the contents being spread out. The more spread out things get, the more easily you can isolate them. Essentially, it's a giant sieve, so to say, and different sized holes mean you get different sized products at different stages in it. And by choosing the type of membrane you're using and a few other factors such as buffers, charges, and so on, you can effectively cause a distributional shift in how things separate out and get either more or less a resolution, so to say. This will bring us to the first kind of blot, and so to say the original recipe, the Southern blot, and it is named after the British biologist Edwin Southern. It came out in 1975, and basically since then it's been used by everybody at some stage if they've been involved in molecular biology. It's used primarily for both the detection and quantification of certain DNA sequences once you have a DNA sample. Now, not only can you use it in that respect, but for example, if you have a PCR reaction where you're amplifying a certain DNA sequence, and that's what you're trying to detect, you can then put this through a blot, or in this case a Southern blot, and by doing that, you're able to figure out whether or not the thing you're amplifying is actually what you think it is. The process is going to be fairly consistent across all of the Southern blot techniques you're using, although there will be some variation within it. First of all, you're going to have to rather obviously get your DNA and isolate it. Now, this can be done fairly easily, and in fact it's not a particularly complicated process. Something like hair, saliva, semen, any bodily fluid essentially. Then you need to break down the various DNA into smaller sections. You want to basically smash it up into tiny bits so that it can pass through that filter membrane and be broken up by size. This is generally done using a variety of enzymes and heating it to about 37 degrees overnight. You then take your gel. Now, one of the common methods here is to use something like agar gel, and you prepare the agar as per the recipe, which is normally about 5% agarose, and you pour this into a mold which has tiny little wells at the end. They look almost like a comb. You then wait for the agarose to solidify, remove the comb, and now you have a well in which the DNA will go. The well and the agarose should now be more or less set, and it should be in a almost bathtub shaped device. You will then add your buffer to this. Now, typically speaking, the buffer should already be added to it, but let's work in sequence for now. After you have the buffer, you then turn on a electrical current, and this electrical current passes through the buffer you're using and carries the DNA along it, because the DNA has a slight difference in its polarity, and therefore, by having electrical current in this, you can pull it along and through the agarose gel. Once you have all of the DNA spread out along the entire length of it, and typically speaking, we're using a ladder on each side to be able to say, this is the size of whatever we found at this particular location, you then proceed with a step called blotting. This is generally done using a sheet of nitrocellulose or sometimes nylon as a membrane, and this is allowing you to remove that DNA section, preferably by either sucking it out or by similar processes. After you've done that, you then have what is, for all intents and purposes, an isolated piece of DNA that you can then use for all sorts of purposes. Whether it's going to be, say, for example, using it in a PCR reaction or something similar. There are all sorts of ways you can use it. To get it out of whatever a particular blotting sheet that you've used, you're going to have to give, other than just having the blotting sheet itself, a product called hybridization. It's essentially a DNA fragment that should run contrary to whatever it is you're trying to extract out of this, and this allows you to remove it and effectively take it out. The process is called hybridization. After hybridization, you can then apply a probe, and the probe allows you to then visualize this. Now, depending on what exact method you're using for visualization, it could be something like a fluorescent probe, in which case you only require light, or it could be something radioactive, X-rays, and so on. The next kind of blot is the north blot, and chronologically it was the next developed. This was developed in 1977 by James Elvin, David Kemp, and George Stark at Stanford. Unlike the southern blot method, this is a little bit different because you're not looking at DNA, or rather you're looking at RNA. But again, you're essentially looking at something that's been extracted from whatever it is that you're going to be testing. So whether that is, say, a tissue from a cell culture, a animal, a sample from a human, biopsies, all sorts of things can be used this way. But remembering, you're looking at RNA and not DNA. Now, RNA is only going to show you what's being expressed in the cell. Effectively, what genes are active, rather than just what genes are present, because not all genes are necessarily going to be active, and a lot of the time whatever's being expressed may not necessarily be expressing a product either. For example, rather than mRNA, you might be looking at long non-coding RNA or other forms of RNA. Now, the end result is more or less exactly the same. That is, you don't need to do a lot of different, let's say, steps. You do need to do some similar things, but you overall have very similar processes. That is, you have to isolate it, you need to make sure you've got at least relatively pure RNA, and then you put the RNA through a gel. The gel electrophoresis is a process that is more or less the same as what goes on with DNA for a northern blot and a southern blot. There are some differences, but for now we'll skip over those. Much like with the southern blot, you blot this, we're going to be saying that a lot in this video, and it gets passed on through a nylon membrane through, say, a capillary or vacuum blotting system. This should leave you with what is effectively the RNA that is present and active in a given sample you've extracted it from. The way that you do this is very similar in some respects to what occurs with the northern blot and the southern blot. The difference with the northern blot is that rather than relying on the hybridization of a DNA strand, you're instead having to rely on an RNA strand that does basically the same thing. The probe essentially does the same job, it's just, well, rather than being DNA, it's RNA. After you've isolated your particular RNA sequence, you can then put this into either a reverse transcription or a real-time PCR reaction to figure out how much and how actively it's being expressed, at least relative to other things. Again, you can also use a latter as well to do so, at least in terms of the gel electrophoresis process. Prior to running any PCR reaction or anything of the kind, you probably want to have a look at it to make sure of the quality and quantity. Now, there are two ways to do this. First, we can use something like ethidium bromide, which is very dangerous and toxic, but very reliable for this, and you essentially use UV light and you can get a reaction and see how solid the bands are. And conversely, you can use something like a nanodrop, which analyzes the certain ratios of different wavelengths of light to give you an idea about how much contamination there is. Unlike the southern blot, it's worth reiterating that you can use either DNA or RNA, although typically you'd want to try and go with a matching process if you want to sort of avoid any issues if you can deal with it that way. But either RNA or DNA can be used for the northern blot, whereas the southern blot you generally stick with DNA. Further to that, you have other ways to be able to figure out what's going on. Like, for example, the previously mentioned ladders and the ability to use UV light or similar to get a certain amount of fluorescence and be able to figure out the size, or at least rough size, of any products that you've managed to get out of your sample. And of course, the final application is that you can create complementary DNA, that is, DNA that matches to your RNA sequence of interest, using this method as well. And this is particularly useful if you're looking at various radioactive isotopes, as you can essentially create probes that are matched with this, and then use something like alkaline phosphate or similar products to get a nice clean image. Unlike the other methods we mentioned where you're reasonably limited in what you can do, not entirely, but certainly by comparison to other methods, northern blots are far more efficient in terms of lab reagent use and sample use. You can get a lot more information and be able to explore a lot more. You can compare things like, say, tissues and organs, different times of development, and so on. All these sorts of things using relatively little sample. Understand, you're really only able to draw a comparison between internal expression, but you can draw certain results out of this. For example, if you have one group of rodents who have been given no treatment, they're your control group. Now you take your, say, a skin sample, muscle, heart, or brain, whatever it happens to be out of them, and that sample then has the RNA extracted, and you repeat the same process for your treatment group. Well, you can look at the treatment group and the control group and go, within the control group, we know that there's this level of activity of this gene, and compared to this control gene or housekeeping gene, it does this. And then you look at the same housekeeping gene in the treatment group, and you look at the ratio of activity of whatever your genes of interest is, and you can figure out, well, in our treatment group, the housekeeping gene was more active regardless, so we know that the rough ratios between groups are going to be this. And of course, you can compare inside group as well, so you know that the rough comparison can be done in-group, intra-group, and inter-group. This is particularly useful if you're particularly looking at gene expression ratios. That is, if you want to see if a particular gene is active in a particular way or in response to a particular thing. So, for example, is there a certain stress response to, say, high blood pressure, to exposure to certain light conditions, food groups? If you want to do the Seralini bullshit, give them glyphosate along with GMO food and mix it all together because that was a mess, and see if the gene transcription, the mRNA, is different between them, and it probably won't be. The final formally recognized blot is the Western blot. It was given this name between two different groups in roughly 1981 and 1979. Strictly speaking, it's been described as independently invented by James Renard, Jacob Risa, and George Stark at Stanford, in 1981 by Neil Burnett, and in 1979 by James Renard, Jacob Risa, and George Stark at Stanford, and Harry Tobin, Theophil Steinheim, and Julian Gordon at the Friedrich Nietzsche Institute in Switzerland in 1979. Ignoring for now the politics of who invented what, when, where, and how, the Western blot is a very similar process to everything else you mentioned so far, which shouldn't be that surprising. Most of this is fairly common, and it's all relatively derivative of the Southern blot technique. You have your gel-type structure and electrophoresis, that is the electric charge that allows it to draw various things down along that current. Whereas the previous two we spoke about have been looking at DNA and then later RNA, this time we're looking at protein in particular. Now, the difference between this method and the others is that rather than using agarose or similar, we're largely looking here at a membrane. Now, the difference is mostly just that, but it does allow for certain other steps to be taken after the fact that are somewhat more useful. For example, immunostaining, to be able to visualize things without having to rely on some of the more, let's say, dangerous chemicals, like ethidium bromide. First of all, you have your membrane. This is going to be in a solution of SDS-PAGE, which is a fairly common electrolyte-type solution, and you're going to have your sample added to this, to the membrane, not to the buffer solution. The reason for the buffer solution is that, like with the others, you want to try and neutralize any particular charge based on the nature of the things you're looking at. So, for example, we mentioned how DNA tends to have a slight positive charge, and so the negative charge draws it down the gel. It's the same thing with a lot of proteins. A lot of proteins have a particular charge to them. SDS solves that issue. But you can then counter that through the electrophoresis process. You then rely entirely on the size of the proteins to separate them out. That is, bigger and more charged proteins tend to hang back more, smaller and less charged proteins tend to get pulled through more easily. This leads to the separation of the proteins in question, and you can then visualize them, for example, using antibody-based fluorescent stains or other kinds of stains. Antibodies tend to be better for this because you can make them fairly specific to whatever you're trying to target. So if you want to, say, find a particular protein, and you want to know if it's being expressed in a particular situation where you have this set up, you can stain for that particular singular protein, and if it doesn't show up at all, either you've stuffed up your antibody, or it's not being expressed at all, and it's basically one of those two scenarios. Unlike the other types of blottings we've described so far, Western blots are arguably a little bit more involved, mostly because you're having to not just extract protein, but then reasonably well purify it down to just the protein you're after, and then ideally try and ensure you get everything out of the cell. Ideally, some sort of lysis buffer, that is something that's going to help burst these cells open, is useful in that respect. However, it's worth remembering that once you've lysed the cell, you essentially are running on borrowed time. You need to have something that will prevent protein degradation for this particular kind of blot, and thereby prevent your proteins denaturing or somehow otherwise interacting and not working as intended, or worse yet, creating something new and wonderful that you don't want. After this, the process generally follows the same process as everything else, but it's ensuring that you get that process of extracting and isolating just the proteins right that is hard. However, once they're all loaded into the wells and the electrophoresis process is running, and typically let's say for half an hour, you're able to get pretty straightforward results because you get the same sort of banding process and results as you would with the other kinds of blotting. If you want more detail on exactly what's going on here, we have an entire video on the isoelectric point that explains a lot of what they're doing when we talk about these particular kinds of Western blots. After the gels have been run, you then proceed to ensure that you've marked everything you need to, and typically speaking by staining it. This is done after transferring it onto something that can be stained. Then after staining, you block it, and then you essentially have it set. After that, you can then provide things like, say, antibody staining. Now, this can be done with the primary and secondary antibody, which is a better way of getting a more accurate and specific result, especially if you're worried about some sort of cross-reactivity or a certain lack of specificity in your antibody. Sometimes this is useful, other times it's not necessary. And you'll find details on the sort of staining process that you'd use in the description box below and linked above. Finally, we get to what we've been hesitant to call the Eastern blot. It's a blot because, well, we've been hesitant to call it a Eastern blot because it's not a single monolithic or homogeneous set of methods or techniques. In fact, it can refer to more than 10 different things, and the time frame, place, and so on that these have been named, the Eastern blot, has varied drastically. The other reason we're afraid to call it that is that we're worried that J. Edgar Hoover might jump out of the closet and arrest us for being a communist sympathizer. And actually that is a legitimate concern with this. One of the major reasons why the Eastern blot doesn't exist as a, let's say, a standard thing is that at the time it was first brought out, there was the Red Scare. That is, people were panicked that people would support communism and communism would spread and ruin the world. And while it has spread, it's only somewhat ruined the world. There are other problems that are probably more pressing than communism. In broad terms, the Eastern blot is a biochemical technique that's very similar to the others we've mentioned, but what it targets is things like lipids, phosphates, and glycoconjugates. Most of the time, and we say most of the time because remembering there's a lot of different variations on this, it's looking at the parts of a carbohydrate that are most useful, particularly talking about antibodies. The first of these methods came out in 1982, and it was, let's say, how to put this politely, it was basically a building on the existing research, and again looking primarily at carbohydrates. In 1984, there was another attempt, and this time it was looking at poly-A RNA, which to an extent is already dealt with the previous methods if done correctly, but this was a specialized variation to make it specifically to poly-A RNA. In 1996, the Eastern Western blot was named, and it looked at the phospholipids and particularly polyvinyl fluoridor nitrocellulose membranes used in this particular application. Then we have in roughly 2000 the Far East blotting, which was made by Japanese researchers, and it was using primarily lectin and antibodies to look at lipids. 2001, we then have glycoconjugates using the BSA. 2002, we have proteins blotted using polyvinylidene fluoride membranes. 2005, we have more proteins. 2006, we have more proteins. 2009, there was a renaming, which was a pain in the ass. which again basically did exactly the same thing previous work had done. 2009 again, we have more proteins, and 2011, another attempt. And given that this is now 2005, you can see that for more than 40-ish years now, they've been trying to get an Eastern blot, but there's been no successive attempt to get one particular technique or method recognized as the Eastern blot, at least not compared to the North, South, and West. As you can see, the name compass of molecular biology may not be a misnomer given how it covers all the major directions of research. Sort of anyway. You have proteomics, genetics, and more. Now, you can look at the Eastern blot included in that and then say, well, there's carbohydrates, lipids, and so on too, which would cover everything you mostly need to worry about. But even if you were to ignore the Eastern blot techniques, it's a fairly good argument that it can stand alone and hold its own in most research if you use the combination of techniques. That is, you can use it in isolation, but if you work with other techniques, and certainly much more involved methods, you're able to further refine your findings and go beyond the north, south, east, and west of blots. But even by themselves, they can yield not just valuable, but vital information when used well. And well is perhaps the key there, because without the well, you don't get much progress with your blotting. Thank you for watching this video. If you have found this interesting, consider liking, sharing, and subscribing. And please do post any comments, questions, or suggestions that you have below.